Hello, I will be discussing how DNA point mutations can be analyzed by comparing the amino acids they produced. Here is a practice coding and template strand of DNA. We will unzip the DNA, focusing on the coding strand and the messenger RNA, or mRNA, that it produces. The amino acids produced start with the codon AUG for methionine. We can use the coding dictionary for UGG to produce tryptophan, AUU to produce isoleucine, and ACU to produce threonine. Then comes AUG, which is a stop codon, and the end of the amino acid chain. With the wild type, we can see what changes a mutation in a single nucleic acid base, known as a point mutation, can cause in the amino acids produced. The simplest mutation is where one base is substituted by a different base, and the new codon creates the same amino acid. For example, the wild type has AUU that codes for isoleucine, but the mutated has been changed to AUC. AUC also codes for isoleucine, so the mutation is silent. We can determine if this mutation is a transition or a transversion. A transversion occurs when a purine, A or G, is replaced by a pyrimidine, C, U, or T, or vice versa. This silent mutation is an example of a transition since the pyrimidine U has been changed to the pyrimidine C, and the other transitions could be purine to purine. The next type of mutation involves changing from one amino acid to another, known as a missense mutation. In this mutated strand, the AUU that codes for isoleucine in the mRNA wild type has mutated to AGU. The AGU codon will code instead for serine, causing the amino acid sequence to change for one amino acid, potentially changing the resulting protein's function. In addition to being a missense mutation, since a pyrimidine U was changed to a purine G, this is also a transversion mutation. If a mutation causes a stop codon to be produced, this is known as a nonsense mutation. For example, in this mutated strand, the UGG of the wild-type mRNA has changed to UGA. The mutation of G to A will be a transition since it is a purine to a purine. More importantly, the resulting amino acid will only be the methionine and then stop, which will almost certainly result in a non-functioning protein. The final point mutation is called readthrough, which is where a mutation occurs in the stop codon, causing it to be changed to another amino acid and the sequence continues beyond its wild-type termination. For example, in this strand, the UGA of the wild type has been mutated to CGA to produce arginine in a transition mutation from U to C. Without this stop, the amino acid threonine and a stop codon would be produced. The additional amino acids may impact the function of the protein. Overall, mutations in single bases can cause minimal or huge change in amino acids produced when compared to the wild type.